In the last few lessons, we've looked at a few different biological processes that humans use. So we've looked at different enzymes. So that was our restriction enzymes, our polymerases, and DNA ligase. We looked at the polymerase chain reaction, or PCR, which is used to amplify or make copies of DNA. And we looked at gel electrophoresis, which was using those gel baths to separate pieces of DNA based on their size. Today we're going to go through this process, which kind of sums up or uses a lot of the things we've already looked at together. And then in the next lesson, we're going to have a video lesson and actually talk through a few of these things together, because I know often there are a lot of questions that come up or things can get a little bit jumbled. So that's what we'll be doing in the next lesson. So today we're looking at bacterial transformation. I have again attached the slides here because there are a lot of images. So if you've got access to a printer and you're taking paper notes, it'd be great if you could print some of them out, or if you're doing virtual notes, you can take them from the slides. Okay, there's a few steps to this lesson, so hopefully we can get through it all in time. A lot of these words are going to look a bit unfamiliar now, but we'll go through it all. So we're going to look at the steps of creating a recombinant plasmid vector. Then we're going to look at the steps of using this plasmid vector to transform bacteria. And then we're going to look at why we use antibiotics in this process. And then finally, how we kind of switch on this bacteria to create proteins. Now, at the moment, you're probably thinking, what is she talking about? Because I haven't actually really explained what this whole process is about. Normally in class, we would actually do a prac of this. Obviously, this year, I'm not sure we're going to be able to do this, but hopefully you can still understand the process without doing the prac. Just a bit of a recap here. So this whole lesson looks at using bacteria to produce or manufacture proteins. Okay, so this is a process that scientists use where we basically use bacteria to make proteins that we might need for medicine, for example. So some really basic theory behind this that you should remember. Firstly, bacteria are single-celled organisms. So one bacteria is an organism, and they divide quite quickly through binary fission. Okay, so remember, we get bacteria, they go through this process and they split into two bacteria. It's a type of asexual reproduction. They don't need a partner to do this. Inside bacteria, they have their normal bacterial DNA, their circular DNA, as well as these smaller pieces of DNA called plasmids. So if this is a bacteria here, this is the circular chromosome that the bacteria has. And then they have these other bits of DNA called plasmids. We're going to look at these quite a lot today. And then another bit of theory that relates to this is that, remember, in DNA, we've got a bunch of genes, which is the same in bacterial DNA, genes that produce proteins when expressed. And by expression, we mean we go through transcription, which is going from DNA to mRNA, what we looked at the very beginning of the year, and then translation, when we go from mRNA into a protein. Okay, this happens in any living organism that has DNA. Okay, so it's the same in bacteria as it is in humans. And that's our little image of that there. Now, using this theory, scientists can use bacteria almost as these little protein-making factories. And I'm going to use the example here of, let's say we need to produce insulin, which is used to treat diabetes. Okay? The way we would use a bacteria to produce insulin is we would first remove the gene that codes for insulin from a healthy human. Okay, and remember, we remove bits of DNA by using restriction enzymes. Okay, so let's say this is a healthy human cell and they've got the healthy insulin gene in there. A restriction enzyme would be used to cut a section of that out. Then this gene is inserted into a plasmid vector. The vector basically means that it will be a carrier. So the plasmid, like we saw in the last slide, is that circular DNA found inside a bacteria. Okay, so this gene gets inserted into a plasmid. So that was the gene there. We've put it inside a plasmid, and now we have a recombinant DNA. We've kind of made a new plasmid by putting this gene in there. This plasmid is then inserted into a bacteria. Okay, so we've inserted this plasmid. There'd be the other plasmids that this bacteria naturally has anyway. We've put a new plasmid in there, and we've turned this into a transformed bacteria. We then let this bacteria divide. Okay, so through binary fission, we now get all of these different copies. This is now, imagine we're in a petri dish, is what this will be done in. So we've made lots and lots of copies of the bacteria, and because each of those bacteria has this plasmid in it, and that plasmid has this gene in it, we've cloned that gene. We've made lots of copies of that gene, and then we can get the gene to be expressed. So to go through transcription and translation to make lots of copies of the protein, which in this example is insulin. So in each one of these bacteria, we switch this gene on, and then it makes these insulin proteins, and then we can extract those proteins and create a vaccination or an injection of insulin, which could be given to a person with diabetes. Okay, that is an overview of this entire process. We're going to go through each step now. This picture I think is quite good. It shows all of it, everything we're going to go through today in sequence. So I think if you can get a copy of this picture, it is great. We break it into kind of three main sections. So this top section up to here is the steps of creating a recombinant plasmid vector. Okay, that is step one, where we end up with this plasmid here. We've put the gene of interest into a plasmid, and now we have a recombinant plasmid that has that gene in it. Okay. Step two, which is this section here, is bacterial transformation, which is basically putting this plasmid into bacteria. And step three is cloning the gene by allowing the bacteria to divide, getting heaps and heaps of copies of it, and then actually producing the protein. Okay, three really, really important steps. Just to note now on plasmids, and you will have to pause the video in a minute. I'll explain what you're going to do, but in my quiz, I've got some questions related to this. Remember, plasmids are bits of DNA that are found in bacteria. There are lots and lots and lots of different types of plasmids. Different plasmids will have different genes and will have different recognition sites for specific restriction enzymes. This is really important because if we are going to insert a gene, let's say we want to put a gene into this plasmid, we need to be able to cut the gene and the plasmid with the same restriction enzyme. Remember, we looked at restriction enzymes and how they cut with these sticky ends, and the sticky ends will only line up with each other if they match, if we've got the same complementary bases on either side. Okay, so we need to use the same restriction enzyme for the gene and for the plasmid. Okay, so scientists have these maps of plasmids, or plasmid maps, and they will select a plasmid that has all of the things that they need for the specific gene of interest that they're going to use. What you really need to know about plasmid maps is how to read them, because this has come up in exams before. So this here is an example of a plasmid map, and I'm just going to go through the key things here. So the plasmid name, the name of the specific plasmid is this bit here. Okay, so in this example, it is PBr322. You do not need to remember the names of any plasmids, but you could be given a plasmid map, and from that you have to identify the name. Okay, so it's that bit in the middle. Then on the plasmid map, it tells us the size of the plasmid in base pairs. So how many base pairs make up this plasmid? And that is this number directly underneath. So this one has 4,361 base pairs. Then we have the locations of specific recognition sites. And remember, these are the sites for specific restriction enzymes to cut. Okay, so I'm just going to go through one of these, and you'll need to write down the other ones to put into the quiz. So the location of this recognition site, you find it up here. It says here, and it's got a number above it, which means, so the number here is 2295. Remember, the entire plasmid is 4,361 base pairs long. Up here you can see a 0. So that basically means that we start, this is the start of the plasmid for the sake of this map. And if we went all the way around, when we get to this specific recognition site, it is at the 2,295th base pair is where that recognition site is. Okay, so you need to figure out the recognition site for these two here. Other questions that have come up in exams before have asked students to calculate how big would a piece of DNA be if it was cut at specific recognition sites. So if we used a few different restriction enzymes to cut this plasmid into pieces, how big would the main fragment size be? Okay, so I'll do the first example, then you need to figure out the next two. So it's saying if we used this restriction enzyme and this restriction enzyme, what would we end up with when it cuts at this DNA? So we'll find this one first. We'll find the HIND III. So that's up here. So that's saying if we got a restriction enzyme and it cut the DNA at this site. Okay, and then we also put this restriction enzyme in and it cut it at this site. How big would our main piece of DNA be? So we're going to say the whole big piece around here, if it was cut there and there. So to figure that out, you would say 651 bases here minus 29 bases, because that's where this one is. And that is going to give us, sorry, let's get my calculator out, 622 bases would be the size of this piece here, sorry. Okay, so that direct size, we won't go past 0. That piece of DNA would be 622 base pairs long. Okay, I want you to figure it out for if we cut here and here, and if we cut here and here. Okay, so in the quiz you'll need to have your answers to these two and these two that you can put in. All right, so we now know how you can read a plasmid map. What we want to do is figure out the steps of creating a recombinant plasmid. So step number 1, we cut the gene of interest out of the original DNA. So this could be our healthy human DNA using a specific restriction enzyme. So in this picture, the gene of interest is this pink bit, and we've found restriction sites on either side. You'll notice there's a bit of overhang there, that's fine. As long as we don't cut in that gene, that's fine. So we cut on either side of that to remove this section from our host DNA or our human DNA in the first place. Okay, so we've now got a bit of DNA with our gene of interest in it. The second step is that a plasmid is selected to act as a vector. So remember, this is when they would look at their plasmid map, and let's say to cut this gene of interest out of the host DNA, it had to use this specific restriction enzyme. We would need to find a plasmid map that uses the same restriction enzyme. Okay, so we've got a recognition site for the same restriction enzyme. The plasmid also needs to have an antibiotic resistance gene. We will come back to what that means, but we have found a plasmid. It's got that restriction site. What we then need to do once we've selected that plasmid is cut the plasmid at that restriction site. Okay, because remember, a plasmid is a circle, and if we need to insert something into that, we need to open up that circle. Okay, so this restriction site has been cut open, and we now have an opening there where we could put our gene of interest. So this fragment is going to be added into a test tube with that and some DNA ligase. Remember, DNA ligase will go along and it will join up loose bits of DNA by reforming phosphodiester bonds. Okay, so here our gene of interest from up the very top has been inserted into this position, and DNA ligase has reconnected the bonds on either side here to create our recombinant plasmid. Okay, it's a combination of two bits of DNA. It's important to know that when this is done, so like lots of plasmids and lots of genes of interest will be put into a test tube together, not all plasmids will actually take up that gene. A lot of the time, if we open up a plasmid here and then put DNA ligase in, it's just going to join this back up again. Okay, so it's just worth knowing from a scientific standpoint, it's not always successful, which is why lots and lots and lots of plasmids and lots of different genes will be added together in the first place. Okay, so at the end of this, we have what's called a recombinant vector that has our gene of interest inside it. This is those same pictures again. I've just put this in here in case you want to use it for your notes. Okay, so this has ended up going in here, and this is the result. Right, the next step now is that we need to get that plasmid inside a bacteria. Okay, it's important to understand that bacteria have the ability to take in DNA from their environment. If bacteria are surrounded by bits of DNA, that DNA can enter the bacteria. And when bacteria take in recombined plasmids that we've made in step 1, we call them transformed bacteria. So the steps of this is that now we mix together bacteria and plasmids. This is generally done in a little kind of test tube thing that looks a bit like that. We put our bacteria in there and we put our plasmids in there. We then do a technique called heat shock, and that is when you put this whole mixture, this test tube would go into ice water for about 10 minutes, and then we would warm it up again. Okay, that's just called heat shocking something. The reason for that is it increases the permeability of the bacterial membrane. If we want these plasmids to go inside this bacteria, we need this membrane to open up a little bit, to become permeable so they can enter. Okay, so we've done our heat shock. After this heat shock, some bacteria will take in plasmids. Okay? Again, in science, nothing's ever 100% successful. So we do lots, that's why we have lots of bacteria in here and lots of plasmids, because only some of them will actually take it in. Okay, so in this one here, this the membrane has opened up after the heat shock and a plasmid has been able to enter. Over on this side, it didn't work. Okay, just for some reason, the membrane didn't open up, plasmids didn't enter. So we've got some bacteria that have the plasmid in it and some that don't. This is the importance of our antibiotic resistance. Remember before we said that our plasmids, the recombined plasmids, have antibiotic resistance, which means they can survive antibiotics. If we want to get rid of, we only want to keep these plasmids that have these bacteria that have taken in the plasmid. So we want to destroy all the ones that haven't. So if we know that the bacteria that have the plasmid have resistance to a specific antibiotic, we will put antibiotics on the plate of all of our bacteria, and all of these ones that didn't take in the plasmid will die. Okay? The non-transformed bacteria will die because they don't have the antibiotic resistance. Okay, it's a really important note. They don't have antibiotic resistance because they never took in that plasmid in the first place, but because we put antibiotics on there, we kill all of them and we are left only with our transformed bacteria that contain the antibiotic resistance and, most importantly, the gene of interest. Okay, the antibiotic resistance is purely so that we can get rid of the ones that didn't work. The gene of interest is what actually matters. That's what's going to make the protein that we need. So again, just a quick summary of that. We combine plasmids and bacteria in a test tube. We heat shock that mixture, which opens up the bacterial membrane a little bit. Some plasmids will enter, which is what we want. We're getting a transformed bacteria. Some won't work. Non-transformed bacteria, where the plasmids just couldn't get in. They were kind of repelled. We apply antibiotics. That kills these ones because they don't have the antibiotic resistance, and we are left with our transformed bacteria. Okay. The final step then is actually cloning the gene and producing the protein that we want these bacteria to make for us. Okay, so remember, bacteria love dividing. If you leave them in ideal conditions at a certain temperature, they will divide, divide, divide. So our transformed bacteria, remember we've killed off all the non-transformed bacteria by now. Transformed bacteria are left to divide by binary fission. This leaves us with colonies of bacteria that contain the plasmid, because all the ones without the plasmid were killed, which has our gene of interest. So if we've got all these colonies of bacteria and each one of those bacteria has our gene in it, we are cloning that gene. We then have to switch on the gene. Remember that genes they're not producing proteins at all times. Genes need to be switched on. So what we do is we expose these colonies of bacteria to what is called what's called an inducer molecule. It induces something. Okay, this might be a real throwback now to Unit 3, but remember we looked at the lac operon, which showed that if bacteria are exposed to lactose, it will switch on the genes that produce enzymes to digest that lactose. That's called an operon. In the presence of a certain molecule, genes will be switched on. That's the exact same way that these inducers work. Okay, when a certain molecule is present, gene transcription is initiated. A common inducer molecule that is used in this is called arabinose. I'm writing this in here because if you are given an example of an experiment, you might come across this. It's similar to how lactose turns on the lac operon. Arabinose is an inducer that turns on the arabinose operon. Okay, but when this molecule is introduced, the gene will be transcribed into mRNA. Okay, so this gene here has been made into mRNA, and then the mRNA will be translated to make the protein. Okay, so this mRNA now produces these proteins, which was the whole aim of doing this entire thing, is to make these proteins, which, as I said in the beginning example, could be insulin. That's the actual product. And then finally, whatever protein it is that we've made, the protein is extracted and it is purified to be used, whether it's for a medical thing, whether it's to make vaccines, but we end up with this pure protein. It gets purified, it would get put in some kind of solution, and then that might get made into an injection. Okay, so that was the final steps of going from these transformed bacteria into making this actual protein. Okay, so again, summarizing everything we've looked at today, I know there was a lot of information, but we start with getting our gene of interest from a healthy individual. That gene is cut out using a restriction enzyme. We've got our gene here. That's what we want to copy. A plasmid is taken from bacteria. Okay, it's a specific plasmid that has the same restriction enzyme that we used for our gene of interest. The plasmid is cut using that restriction enzyme, so it opens up. Now this gene of interest is inserted into a plasmid. Okay, and we've made a recombinant DNA plasmid with our gene of interest. That was step one. Now that recombinant plasmid is taken in by bacteria. That process of bacterial transformation, where we mix them together, we heat shock it, this opens up and the plasmid enters. We have our transformed bacteria. Those transformed bacteria are left for a few days. They go through binary fission. They make lots and lots and lots of copies. Each copy contains that plasmid that contains that gene of interest that we want to copy. And then eventually an inducer is added and that gene of interest starts producing all of the proteins that we need, which are then extracted. I've just included a couple of other pictures. Different people kind of like different versions of this, so feel free to use these if you want. It kind of shows it more how it would look, you know, if we were doing it in a lab. So we've got our DNA. These are the plasmids here. So it kind of just shows from that step. Plasmids and bacteria are mixed together. They're heat shocked. We open up the bacteria a little bit. Plasmids enter and we get transformed bacteria. Both of these, so remember some bacteria don't transform. All of the bacteria are on a plate and we put an antibiotic there and it kills off all of these non-transformed bacteria, and we are left now with transformed bacteria that produce colonies, and then from there those transformed bacteria produce proteins. To then produce the proteins, they would be extracted into these columns. Don't worry too much about this antibodies binding to the target protein. You don't need to know that. That's just a scientific step in that, but we end up with these pure proteins. All right, I know that was a lot today. See how you go time-wise. If you're getting, if it's been 98 minutes, you're only halfway through this, that's okay. Take a break. Like I said, in the next lesson, I want to do a bit of a recap of all of these things, but I might just wait and see how everyone is going time-wise. If you're really struggling with this, we can push that to be towards the end of the next lesson, so you've got time to catch up on this. So if you are struggling with time, maybe just send me an email. Don't do the quiz until you can actually do it all. Just send me an email and let me know how you're going time-wise. But yeah, we will definitely chat through these things again, because there is a lot of information that we've gone through in the last week and a half. All right, see you later.